Hello everyone, just a few notes on the formal lab report for our acetaminophen experiment. Okay, going back to abstract and experimental. Abstract, just write purpose, brief highlight of the procedure, and the major results and conclusions. So in this case, the purpose, you have that in your lab manual, but for each of your groups, you had sort of a specific variable that you were testing. Whether that was, the effective pH or the effect of different formulations or whatever it was, right? Looking at each of those, the effect of each of those on the rate of release of acetaminophen from, again, whatever formulation or formulations you measured. Again, then you've got that highlight of the procedure. There are a lot of different ways to measure acetaminophen. Sort of briefly describe how you did measure acetaminophen in this experiment by converting it to ferroin, measuring the absorption using, measuring the absorbance using UV vis at a certain wavelength, etc. And then major results and conclusions. Remember that this is going to have sort of specific numbers in it. In this case, there are sort of a lot of numbers, but, again, highlight sort of the key numbers for your project, right? If you were measuring the say different formulations, right? You might need to have, those key numbers that describe, okay, the gel cap released the fastest, and, have the key number for that, maybe that one key number for how fast it released for each of those three formulations, say, or for each of the three pHs. So yeah, again, you can't necessarily get every number in there, right? But maybe like one number that describes the rate of release and maybe one number that describes the total amount released for each of them, right? That's 6 numbers. That's, one for each of your 3 trials. Something like that might be reasonable. Then your experimental. So in this case, this is going to probably be your longest experimental of this semester, or it definitely will be your longest experimental of this semester. Again, try to describe each of those different experiments that you did in, a nice sort of pair. Remember to be specific about exactly what you measured. Measured the absorbance at 703 nanometers. Exactly what instrument you used for your UV vis. But yeah, again, keep it as short as possible here, just describing each of those 3 experiments. Okay, then when it comes to results, again, since you had a lot going on, there's a lot potentially going on in the results section here. Again, you just got to sort of walk the reader through exactly what you did, thinking about, okay, you did these different experiments in order to measure the rate of release of acetaminophen, right? How much acetaminophen was released at each time under these different reaction conditions. So you've got to sort of walk through exactly what that looks like for each of the 3, in each sort of part there. The handout from lab is sort of shown out here, right? Parts 1 and 2 were where you determined lambda max, constructed your standard curve, and did that conversion of a stock solution of acetaminophen to ferroin. So, I don't know what happens here, right? A lot of this is going to be very similar to your cobalt chloride report, right? If you think about sort of why you did this, right? We can't measure acetaminophen directly because it's sort of clear and colorless with our spectrophotometer. But by doing these series of reactions, we can turn it into ferroin. Then the ferroin, with that bright red color, we can detect with UV vis, right? So that was our approach. We're going to detect acetaminophen by first converting it to ferroin and then measuring ferroin with our UV vis. So, right, 1 through 3 are sort of going through, okay, given that that's our approach, we need to have a sort of nice standard curve of ferroin, just like we had for cobalt chloride. Walk the reader through, okay, we're taking this approach of converting acetaminophen to ferroin, so we need a standard curve of ferroin to be able to go between the absorbance that we measure and the concentration of ferroin. Then using stoichiometry, go back from ferroin back to acetaminophen. So that's sort of 1 through 3. again, then we have that sort of test conversion of acetaminophen to ferroin. That was sort of part 1 in your lab notebook, right? That was the case where we took that stock solution of acetaminophen and converted it, right? And again, if everything we're doing is right with all of our math, all of our standard curve, all of the steps that we took, we should get the known concentration of that acetaminophen stock solution out in calculation number 4 here. So again, this is, if you think about sort of why we're doing this, this is giving us a good check on this process of converting acetaminophen to ferroin, right? If we can get within, 5 or 10%, that's a good sign that our procedure, all that is working properly, right? So that's sort of your first, big sample calculation. I guess you can have your dilution here, just, again, similar to the last report. But yeah, you'll want to sort of, use that evidence to say, yeah, okay, we actually can convert acetaminophen to ferroin. So this is all sort of our setup control experiments from that first week of lab, right? Then you have the next set of experiments, which are all about the acetaminophen content and rate of release. Right, so in that case, you've got what we spent most of the time in that data analysis on Monday doing, right? You're going to have all of your different figures right back here for your for this, right? It's useful to also have your the actual pictures of the spectra, right? Again, just like you had for cobalt chloride, you can get some information about ferroin versus is there anything else in that solution there back in parts 1 and 2. Okay, a lot of calculations in here, right? But what you finally get out of part 3 are your final graphs, right? Here's what's going on with the acetaminophen content and rate of release. You can, explain to the reader exactly how you're able to calculate acetaminophen from the given sort of absorbance versus time data. So you can give the reader that sort of overview of, okay, from the measured absorbance, here's how you go through and determine the milligrams of acetaminophen released. And then, right, you'll got all of your plots here. Sort of get all the information you get off of each of these curves about how much acetaminophen was released, right, at the very end of the curve, what's going on at the end, what's going on at the beginning, right? Linear region is that initial region of the curve, right? That is what's going to let us estimate the time it takes to release 100 milligrams and half of the total. So you'll show those calculations. And right from this sort of release amounts, right, the formulation, how much is released, and the time it takes to release 100 and half, we can get some idea of finally answering our purpose, right? From that idea about how much time it takes, we can see which one was sort of releasing the fastest, which one was releasing the slowest. Was there no difference? Yeah. And again, any of the sources of error, right? How far off are you in terms of percent error, right? The best way to summarize all of that data from the graph is is your nice table there. So yeah, again, when you're thinking about drawing conclusions, you can definitely use the class data to supplement where it's useful. And again, if you've got control experiments, those are also really good to sort of include and analyze and sort of show, okay, we did this control experiment, here's what's going on there. That's really useful for your discussion section, right? So you've got a nice data table for your overall lab reports here. Yeah, probably one, maybe 2 nice tables would do a good job of including all that information so you can actually draw conclusions. Discussion, right? So we've got just sort of, qualitative, what's going on with the different acetaminophen contents, right? That sort of gives you an idea of, did you overestimate, did you underestimate, did you fully release the acetaminophen or not? Drawing those conclusions, again, that's where the class data here could definitely be useful as something to compare to, right? If you look on the D2L site, we have all of our class data here. So the combined class data here. you had 3 trials that you did, right? But there's all of these trials from prior years that can be really useful, right? So here's all of the combined class data for formulation versus time at the normal pH of 1, just using the 0.1 molar HCl. We've got a lot of class data here. You can, again, you could just include this figure as is if it's relevant for you. That's right here. Download the plot as a PNG again. Just a good sort of check on what you did, that your conclusions from 3 trials, hopefully, you can use this to get more robust conclusions. You don't want to sort of overinterpret your data. You can see there's a lot of error on these. Even after doing 9 trials, there's still a lot of uncertainty because of all those different variables, the stirring, all the different steps in doing the conversion. So you don't want to overinterpret your results, but you can sort of see there are statistically significant differences here between the formulations, especially when we combine, in this case, 2 years worth of class data. So yeah, that's sort of the idea of number 2, really your biggest point of really drawing conclusions related to the purpose of the experiment. Then a little bit more about possible sources of error, right? I've got 4 different options here. Again, I would lean towards things that you have relevant data on, right? If you did control experiments that relate to any of these, that would be useful to use that one. If you saw a big overestimate in the amount of acetaminophen, right? If you consistently got 600, 700, 800 milligrams of acetaminophen when there should have only been 500, maybe you want to think about what are some of these that would cause you to actually overestimate it. Number 4, future experiments, right? Don't just propose more trials of the exact same thing, right? I mean, I think the idea there would be, are there other variables you'd be interested in varying? Are there other of these control experiments that you could do, right? Is there something you could do to actually address any of these sources of errors? This is definitely a little bit harder, but not a bad sort of way to conclude, sort of thinking about what room there is for the future, right? Again, there's a ton of variables in this experiment. A lot going on. Is there some area that it seems like you could think about doing, more trials in that's not just the exact same thing we did before? Again, if you've got any questions, definitely I'm happy to look over that draft of some or all of it if you send it to me by roughly around 5 on Saturday. Otherwise, yeah, good luck writing your report, and see you on Monday.